First, we are going to talk about DNA replication. DNA replication begins at the origin of replication. Helicase unwinds the DNA strand. Each single strand is held in place by single-strand binding proteins. To prevent the strands from overwinding, topoisomerase rejoins DNA strands and relieves the strain in a double helix. If the strand runs from the 5' end to the 3' end, it is known as the leading strand. DNA primase comes in and puts an RNA primer on the beginning of the leading strand to start the process of replication. DNA polymerase III comes in after the primer and adds on the complementary nitrogenous bases to the leading strand. It can only run towards the 3' end. The lagging strand runs from the 3' end to the 5' end. This is where the DNA polymerase III starts its copying process and moves backwards towards the 3' end. This is why this process occurs in segments again and again. These sections are called Okazaki fragments. DNA polymerase I replaces the RNA with DNA, adding nucleotides to the end of the fragment. DNA ligase fixes the gap that is left behind so the strand is continuous and no longer in fragments. This is the semi-conservative model of DNA.
Transcription is a process in which RNA is synthesized from the DNA template in the nucleus. The DNA contains a promoter and a terminator which signal the beginning and end of the sequence to be transcribed respectively. The promoter in eukaryotes is known as a Tata box because it contains many A and T nucleotides. Transcription factors, a collection of proteins, bind RNA polymerase II to the promoter site to create the transcription initiation complex. Then, the DNA double helix is unzipped, creating what is known as a transcription bubble. The RNA polymerase II reads the DNA in a 3' to 5' direction, attaching complementary RNA nucleotides to the DNA strand until it reaches the terminator. It is important to note that in RNA, the nucleotide uracil is used instead of thymine. Once RNA polymerase II interprets a terminator sequence, the RNA molecule is released and the promoter detaches. In RNA editing, the pre-mRNA that resulted from transcription is modified before it leaves the nucleus as mRNA. Pre-mRNA is made up of introns, non-coding segments of the nucleic acid, and exons, coding segments of the nucleic acid. In RNA processing, each end of the pre-mRNA is changed. A 5' cap is consisting of modified guanine nucleotides is added to the 5' end, and a poly A tail consisting of repeated adenines is added to the 3' end. The poly A tail also serves to allow the RNA strand to pass through the nuclear membrane and into the cytoplasm. A spliceosome containing small RNAs and proteins removes the intron and connects the exons back together, creating a final mRNA ready to enter the process of translation.
The mRNA, after leaving the nucleus, comes to the ribosomes in the cytoplasm. The ribosomes are made up of a large subunit which has the E, P, and A site and a small subunit that has the mRNA binding site. The mRNA then goes in between the large and small subunit and attaches to the small subunit upstream the start codon. In initiation, the tRNA brings an amino acid which is determined by the anticodon of the tRNA to the AUG start codon of the mRNA which codes for the amino acid methionine in eukaryotes. Next, the large subunit binds to create the translation complex, completing initiation. In elongation, a tRNA binds to the A site, forming a peptide bond between its amino acid and the amino acid of the tRNA in the P site. The translation complex then slides down one codon, shifting the sites the tRNA are in. The uncharged tRNA now in the E site exits the large subunit, and the elongation process will continue over and over until a stop codon has been reached. At the stop codon, a release factor binds in the A site and the polypeptide chain that has been created is released from the tRNA in the P site, and the whole translation complex disassembles and translation ends.
Operon is a functioning unit of DNA containing a cluster of genes under the control of a single promoter. These are usually present in prokaryotes. In eukaryotes, each gene has its own promoter. An operon consists of 4 parts. The promoter, which is the binding site for the RNA polymerase, the operator, which is the attachment site for the repressor, which attaches to the operator and keeps the RNA polymerase from attaching to the promoter, and the cluster of genes. In an inducible operon, the presence of an inducer, in this case lactose, will change the shape of the repressor. This will remove the repressor and allow the RNA polymerase to attach and start transcription. The mRNA will go code for proteins, which will come back and remove these inducers. The repressor will then change shape again, attach to the operator, and shut down the operon once again. In a repressible operon, the lack of the metabolite, in this case tryptophan, will change the shape of the repressor. This will remove the repressor and allow the RNA polymerase to attach and start transcription. The mRNA will code for proteins which will come back and create more tryptophan. This tryptophan will attach to the repressor. The repressor will then change shape again, attach to the operator, and shut down the operon once again.